Okay, so we're gonna start with talking about whoops, start talking about immunity here. So the last thing was about innate immunity. So now we're going to talk about acquired. So acquired is also kind of like adaptive. So this kind of splits into two categories rather. So active is when we make our own antibodies. So that's when we're exposed to an infectious agent. So say you get exposed to chickenpox. So then you make your own antibodies to it, and that helps protect you against it as you age. Artificial active is when you get a vaccine and then your body starts to make antibodies to it. So then that protects you. So it's still active because your body's making its own antibodies. And then passive is when you're given antibodies. So it can be natural, which is from the mom. So antibodies can cross the placenta, or they can be transmitted in breast milk, or we can give you antibodies from other sources. So we can give you an antibody injection. So we could give you IgG. Okay, so now adaptive immunity. Shoot, it doesn't look right. Sorry, guys. So adaptive immunity, the first thing I want you to remember, and we've already gone over this in the first part of this, is that an antigen causes an immune response. So typically antigens are proteins. And we kind of went over all sorts of different things can cause an immune response. Okay, now within the antigen, if it's big, okay, it can have several different areas where the antibodies bind, and those are called epitopes. Okay, now an antibody is basically a protein. So it's a protein that is produced in response to an antigen, and it's going to be able to bind a specific antigen. Most of the time they look like Ys. Okay, so this is what an antibody looks like most of the time. There are some other variations. Now, this picture shows how antibodies can bind to different So this is a big antigen, and it's got different epitopes on it. So different antibodies. So the antibodies can bind to a different epitope. And then here's how antibodies can bind to bacterial cell. Okay, so I know what I have down here. I have titer. Okay, so titers, we always talk about titers because we're trying to see if somebody has a certain amount of maybe antibodies to an antigen. So they'll do a titer, like when you're going to nursing school, to make sure, if you don't have vaccine records, they'll do a titer to make sure that you have enough antibodies to a certain disease. So a titer really, the book says it's a quantity of a substance needed to produce a given reaction. So it's the antibody titers, the quantity required to bind to and neutralize a particular antigen. So they know what your titer should be. So they always say they're doing a titer test or doing a titer. So you look, it's used different ways, but it's a test really to see how much is in there. Okay, now immune system compartments. Okay, so we're going to talk about humoral, and we're going to talk about cell-mediated. Okay, so humoral is, I always think of humors. So humors are in your blood or in your body fluids. So humor is body fluid. So we have antibodies that are going to be found in there. Their B lymphocytes are part of it. So we're going to talk about how they develop. We're going to talk about their abundance and what they protect against. Okay, and then cell-mediated are T lymphocytes. Okay, and we'll talk about how they develop, how abundant they are, and what they protect against. Okay, so we're not really going to talk too much about the development. They know a lot. They know exactly how they recognize self, how they develop, and we're not going to spend a lot of time on that. But we'll talk about the basics of them and how they work. Okay, so general properties of immune responses. So recognition of self versus non-self. Okay, so the hypothesis that they call it is clonal selection. So they don't let any clones or copies of cells out if they don't recognize self, because that's how we get an autoimmune disease. If we have something that doesn't recognize itself, it starts to attack itself. Okay, so recognition of self versus non-self. So here's what the book says: the ability of the immune system to tolerate host tissues while recognizing and destroying foreign substances, probably due to the destruction or deletion of clones of lymphocytes during embryonic development. So we don't want anything to get out into your body or into your blood that could damage your own cells. So they go through like a trial period or a test period where we don't want them to respond. We don't want them to attack your own body. Okay. Now, specificity and cross reactions. Okay, so we don't want, when your immune system is working, we want antibodies against chickenpox to just work against chickenpox. We don't want them to cross react with something else. So sometimes that happens, and that can cause an autoimmune disease too. Okay, so cross reaction means it could be reacting with another pathogen, but we worry about it because we don't want it to react with a tissue in your body. We don't want it, if an antibody doesn't recognize an antigen clearly, it can cross-react. So an example of that would be, so maybe rheumatoid arthritis. So we have cross-reaction of antibodies that are being produced, and they start breaking down the joints. Okay, now heterogeneity or diversity. So it can respond to a variety of different foreign antigens. Okay, and then we have memory. So we want to be able to remember or have memory cells so that we can develop a quick immune response. So that the anamnesiate, which I don't know if I said that right, response, which is a secondary response, we want it to happen fast. Okay, and we want it to be specific. Okay, so types of antibodies, and there's this lovely chart, which you're not going to memorize all this stuff on the chart, but I want you to realize that there are lots of different types of antibodies. Okay, so IgG is the one that is the greatest percentage of antibodies that we have. Okay, so IgG we start making after we get exposed to an infectious agent. Okay, now IgM, babies make 6 months after birth, so that's where we have the anti-A, anti-B, anti-AB, so blood typing. Okay, then we have IgA, which is found in body fluids. We have IgD, and we have IgE. Okay, but the big one is IgG. Okay, so they last, those antibodies last about 21 days. Okay, they're going to activate the complement system. They can cross the placenta. They look like the Y-shaped protein. Okay, so now how does an antibody work? So antibodies work typically 3 different ways. So they cause agglutination. So what can happen is they can cause let me see. I'm trying to find a good example of agglutination. I didn't look at what the picture was I put in here, but it's, so agglutination, what happens is the antibodies bind to the antigen, and they can cause it to clump up. And then after it clumps up, it can be engulfed by a macrophage. Okay, so it can also kind of tag something. So that's what this picture is supposed to be. So here's the macrophage. Here's the bacteria. The antibody is binding to the bacteria and it's kind of tagging it. so that it's complementing it. So it's opsonization. So it's tagging it so that the macrophages can actually engulf it. Okay, this one, it's neutralization. So here's bacterial toxins. Okay, and then if the toxins bind to the cell receptor, it's going to cause problems, right? But the antibodies can bind and neutralize the toxin, and then they'll get ingested. Okay, and then IgM, okay, so if you have bacteria in your plasma, okay, it can bind to the bacteria, and then it creates a complex, and then that's going to get broken down. Okay, agglutination is when it all clumps together. Okay, now I'm kind of cruising through this, so I hope everything is, I hope you guys are okay, but I'm trying to get the stuff that I think is big picture. Okay, now monoclonal antibodies are kind of a new concept. Well, they're not that new, but they're now being used to treat a lot of things. So you'll see drugs advertised, and if they end in MAB or MIB, okay, those are actually monoclonal antibodies. So what people are doing is they're making antibodies in animals, and they're very specific. Okay, so they're super, super specific, and then they can be used as a drug. So basically, here's a mouse, okay, and they inject the mouse with the antigen. Okay, and the spleen is where the B lymphocytes are. Okay, so the B lymphocytes, the spleen is removed from the mouse, so the mouse is euthanized. Okay, so the spleen cells are removed, okay, and then they start, they separate out the cells. Okay, so they add some enzymes in it, and the connective tissue of the spleen breaks apart, and they can get the B cells out. So then what they're going to do is they're going to take these mouse cells that could be making an antibody, and they're going to combine them with mouse myeloma cells, so mouse cancer cells, basically. So they take the spleen cells and the cancer cells, and they put them together, and they actually fuse, and they call it a hybridoma. Okay, so a hybrid. So the hybrid is a half mouse spleen cell and half cancer cell. Okay, so then they'll grow those hybridoma cells up, and they're going to try to find ones that will make the antibody that they're interested in. So once they can do that, okay, they can just purify one cell and grow them up, and they can have those cells make antibodies. Okay, and then the antibodies can be produced, they can be purified, and then they can be used for whatever. Okay, so they could be used for diagnostic benefits. Okay, so they could be used to try to identify a disease. So instead of using something like they could be used kind of like a pregnancy test, but a pregnancy test is not as specific. These would be really, really specific. Okay, they use them to treat cancers now, so that they're very, very targeted. So if they can make the antibody to a certain part of the cancer cell, they can tag it and hopefully stimulate the patient's own immune system in order to break down the cancer cell. Okay, so kind of makes sense, I hope. It's the future. It's a big, huge industry right now. Okay, challenges of it would be is if you have cross-reactivity. So if your body starts to react to the antibodies, if the antibodies aren't specific enough, keeping the antibodies in the body long enough. But they've actually come a really, really long way, and it's very promising. Okay, now if we talk about cell-mediated immunity, okay, we want to talk about antigen-presenting cells, or APCs. So antigen-presenting cells have a marker on their cell surface. So once they encounter a certain antigen, Okay, what they'll do is they'll present it. So they'll present it to the other cells and they'll basically say, hey, guess what? I killed whatever this little purple thing is here. Okay, now immature T cells can differentiate into three different kinds of T cells. So one is a helper T cell. So helper T cells are kind of like the general of the army. They're going to control the specific immune system. So we call them TH. Sometimes they call them CD4s. There's a bunch of different names for them. Cytotoxic T cells are more like the infantry. So they're going to go and they're going to start killing cells. So they're cytotoxic, so they kill cells. And then there'll be memory T cells. So they're going to remember what you actually see. Okay, so TH cell activity. So if we look at this picture, and this is actually in your book on page 488 if you have the 10th edition. Okay, so what happens, okay, so here's a macrophage, so it's an antigen presenting cell, okay, and it engulfs some sort of antigen, and it presents it in this MH class 2 marker. Okay, so it presents it so that all the other cells in the body know what's happening. At the same time, it produces this other signaling cytokine, which is called IL-1, so interleukin 1. Okay, so the T cell, okay, can bind, has a receptor on it that is going to bind to the antigen presenting part. Okay, and then the T helper cell gets activated, okay, and it produces interleukin 2. So then interleukin 2 is going to stimulate these other things to happen. Okay, so we're going to get another T helper cell so we can keep spreading the message. And then we'll have T helper cell 2, which will activate B cells so we can start making the antibodies. And then we'll create memory T cells. Okay, now the T cytotoxic T cells, they're going to be activated with a different receptor. So it looks different. The receptor on their cell is different. But what will happen with it is, so it'll get activated, okay, and then it'll start releasing these destructive enzymes. So basically what it'll do is it'll destroy any cell that might be abnormal or infected by a virus. Okay, so they make, I was going to write it down, but they make a protein called perforin, and perforin perforates or puts holes in those cells that have that don't look normal. Okay, now what can modify an immune response? So there's a whole laundry list of things that can change your immune response. So it could be genetic, okay, and age. So either babies that haven't been exposed to a lot, or elderly who are weak. Okay, so the season, diet, so whether you're eating healthy or not, whether you're exercising, so the more your lymph moves through your body, the more stuff circulates. If you're sleep deprived, if you're pregnant, trauma, medications, so think about like cancer treatments or things like that, and the environment can all decrease your immune response. Okay, now active immunization. Okay, so when we immunize someone, okay, we're either vaccinating with the organism that's been attenuated or modified, or we're vaccinating with an active toxin. Okay, so the toxin won't be harmful anymore, but it creates an immune response so you can make antibodies to it. Okay, so we don't always use the organism. Sometimes we're using the toxin. Okay, now here's recommended immunizations, and there's actually charts in your book, a whole bunch of charts in your book about immunization and when you should get them and what's contraindicated with them. So it's table 18.8b is in there. Okay, so DTaP is diphtheria, tetanus, and pertussis. Okay, so diphtheria is a respiratory disease that's pretty much eliminated, but when they stop vaccinating, it goes up. Tetanus is a clostridium, so the toxin causes muscle paralysis, and pertussis is whooping cough. Okay, and then MMR is measles, mumps, and rubella. HIB or HIB is I don't know, Haemophilus influenza type B. I think that's right. Why do I always mess that one up? I think that's right. Yeah, Haemophilus influenza type B. Then HPV, so human papilloma virus. Okay, so hazards of vaccines. Most of the time, there's just maybe an immune response. So people have soreness at the site of injection. They might have feel a little bit yucky. But there are lots of instances in the past where companies have messed up or had didn't do a good job making vaccines. So there are some instances of bad vaccine manufacturing. There haven't been any in a while, but there were definitely some problems before. Sometimes people have an immune response. So if they have an adverse reaction, then you need to make sure you tell your doctor so that they don't get it again, because it seems like it gets worse every time. But okay, so page 499 has a couple paragraphs about hazards of vaccines. Okay, passive immunization would be if we gave you the antibodies. So antisera, so if somebody has a bite or like a venomous snake or a spider bite or something, they might give them antisera. Sometimes they give IgG for things too. Okay, so like diphtheria, botulism, rabies, they'll give you IgG against that toxin. Hyperimmune sera. Okay, so this is serum that's been that has. Well, now I'm looking for a hyperimmune sera, or it's got a high concentration, so it's antisera that has a high concentration of IgG. So it's something that's kind of been beefed up. And then antitoxins are going to be antibodies to toxins. Okay, sometimes they consider monoclonal antibodies passive immunization, especially against RSV. Okay, now let's see. So future of immunization. So you need to remember these are the things that they want in a vaccine. It should be protective. It should be safe. It should last. It should generate active immunity, and it should be stable so that you can get it out to different people. So you could have a whole cell killed. Okay, we could have attenuated. It could be a subunit. And now they spend a lot of time working on recombinant DNA vaccines. Some things at times it's just really hard to get a vaccine to work. It's hard to develop it to certain viruses or certain pathogens.